Hello again. In the previous video we talked about the use of the calcium alginate for encapsulating the cells, and we told you that when we have the suspension of the cells and the alginate here and we mix them together by injecting that into this bath containing the calcium chloride, when these droplets are introduced here due to the presence of the calcium, which are divalent, the self-assembly of these polysaccharides will form the gels. But the size of these droplets should be decreased as much as possible to provide much more surface for the exchange of the nutrients and their products, the therapeutics which are produced by the cells. So the higher surface area will be better for us. So we want to reduce the dimension of the droplets which are going to fall down here, and in order to do so we can use the air inlet to reduce the size of the droplets or the high voltage. And in another strategy, a sort of a cutting tool can be used to cut the droplets when they are coming out of the needle, so that their size is decreased. Let's continue with this machine. You see here that it has a nozzle, and by having the mechanical vibration, the vibration of this nozzle will destabilize the laminar flow of the alginate which is coming out of this machine. And if we have another bath here containing the calcium, divalent calcium cations, the self-assembly will occur and we will form the gel. Now we want to mention a drawback about this strategy. If you remember, we mentioned that in the matter of encapsulating the cells inside the gels, we talked about the RGD that I'm going to show you now. And we said that the cells need to feel the mechanical force here. So that the presence of the RGD is essential so that the cells feel the mechanical force between each other, so they will survive. But for the calcium alginate, the problem is that we cannot introduce the RGD, so the cells cannot survive. So what should we do? Instead of introducing just one cell, we will encapsulate the cell islets. You saw it here. I'm sorry that I'm going up and down. I just want to show you that we said we will encapsulate the cell's islets. And what are the islets? Islets are not just the one single cell, is the population of some cells which have grown and their cellular membrane has been already developed and they are connected to each other so that they feel the mechanical force already. So we are going to encapsulate a population of the cells, but there is a drawback with this. Consider this as a population of the cells. If we are lucky, we can encapsulate inside our matrix all the population of the cells, all the islets. But in some cases, some of the islets will be outside, and the problem is that it can be recognized by the immune system of the body, and the consequence inflammation happens. Now let's go to another drawback of the encapsulation of the cells. If you remember, here we told you that we need a semi-permeable, let's say here, a semi-permeable matrix for the exchange of the nutrients and the products, therapeutic products and the waste products and etc. So this semi-permeable matrix we expect to be homogeneous, to have the pores with the same size everywhere, but this is not going to happen easily, although we like it to happen. So we will finally lead into the formation of a gel with an inhomogeneous pores, and the problem is with the mechanism in the formation of the gel. I am really sorry that you see all the notes and everything together, but I try to tell you the concept and the important information about this slide, the pore size. Well, you see that when the gel is going to be formed, first of all, the outer layer is formed, the calcium and the polysaccharides, which will self-assemble and form the outer shell. But for what remains inside, the calcium has to diffuse inside to further form the gel. But this is the problem: the diffusion of the calcium will decrease, so we will lead to the formation of the pores, which are homogeneous at the outer shell, but the more we get inside, the less the self-assembly of the polysaccharides will happen. So overall our material is going to be inhomogeneous, so the alginate bead is going to have inhomogeneities inside that. Another problem with the encapsulation of the cells are the, let's talk about the batch to batch variability. What is the batch to batch? Batch to batch means that if you produce something now and then you produce it another time, you need to have the similarity between what you produce now and what you produce later. So you know that for the formation of the calcium alginate gels, we use the polysaccharides sequences, but sometimes these the sequences of the polymers that we are going to use are the same, but sometimes they are not, so the properties of the gels are going to differ generally compared to each other. Well, the last few slides are not going to be that important. Just let me jump to this slide, and you see here that a concentric nozzle has been used, which contains two materials inside. As you see here, the red one and another one around it. And when the gel is formed, it's like a core share material which can have the cells inside and the good, let's say, situation, atmosphere and environment for the cells to work, and this shell which protects the inner layer from the environment outside. So this was all about the gels and injectable nanomaterials, and for the next videos we will cover the immune system of the body.